Right, welcome back to our second lesson in Unit 4 for cell division. This is chromosomes and karyotypes. There is a note taker to go along with this if you wish. For our objectives for this unit or this lesson, by the end of this lesson, you should be able to do a couple of things. Define what a chromosome is and what it does for the cell. What is its purpose? Label the parts of a chromosome. Begin to understand the changing role of a chromosome in the process of cell division. We will spend a lesson talking about this later, but start thinking about it now. Start making those connections. And finally, be able to understand or read a karyotype. So when we talk about a chromosome, what we are talking about is a condensed version of our DNA within our cells. So chromosomes are structures made up of chromatin, and that chromatin is what contains all of our genetic material, or our DNA. Chromatin is the complex of DNA and protein structures that make up the genetic material. So as you see here on the right, we have our double helix. That double helix DNA is wound around the nucleosomes, which are made up of histones. And as they compile together, they form into a chromatin fiber, all those little squiggly fibers that you see under the microscope when you look at a cell when it's not dividing. That is our chromatin. When a cell is required to divide, that chromatin condenses into chromosomes, the structure you see up here at the top. Human beings have 46 chromosomes made up of two sets of 23, half from each parent. So half from the sperm and half from the egg come together to make up 46 chromosomes. This number of chromosomes differs in various species, but in humans it is 46. So what is the structure of a chromosome? We have the form it usually exists in on the left, and then kind of in the middle, the double helix, the double strand, the two sister chromatids. That is what occurs during cell division. In either form, we have the centromere here at the middle. The centromere here is made up of the kinetochore and associated spindle microtubules. I have here on the right, this is important during cell division, and it is. During cell division, the centromere gets attached to these spindle microtubules via the kinetochore to help during cell division to pull those sister chromatids apart. And then each chromosome is capped off with telomeres. Telomeres are simply strands of the DNA with repeating copies of the A with the base pairs continuing on. This prevents the chromosomes from fusing together at the ends, as this outside region is not used during replication process. It doesn't contain any useful information. So it prevents them from fusing, and it prevents the DNA from fragmenting at the end. Each time a chromosome divides, this telomere shortens a little bit. When we talk about karyotypes, what we are talking about are pictures of a set of chromosomes that help determine certain genetic information, not all. Karyotypes are taken of somatic cells. So when we talk about a somatic cell, what we are talking about is a cell in our body, such as muscle or skin, something that contains a full set of chromosomes. We are not talking about a sex cell. This is a gamete and only contains half of the set of chromosomes. When we want to look at a karyotype, we want to look at the full set of chromosomes, all 46 in their diploid state. The sex cells exist in a haploid state, only 23 chromosomes. Karyotypes also group our pairs of chromosomes. So as you can see here, we have a karyotype on the right. It has all 46 chromosomes grouped in pairs. Most chromosomes are autosomes, chromosomes that do not relate to the sex of an individual. So 1 through 22 are autosomes. The X and Y chromosomes are allosomes. These are chromosomes that do affect the sex of the individual. So take a second, guess the sex of this individual based on what we see. If you guessed female, you are correct, as they have two X chromosomes. A male would have an X and a Y. So what can karyotypes tell you? Because this is just a picture of our chromosomes. It doesn't tell us what those genes say, it doesn't tell us all the genetic material on them, but it can tell us certain things. It can tell us the sex of the individual, male or female. Are they XX or XY? The number of chromosomes, it should be 46, but there are cases where it is not. Sometimes you have multiple sex chromosomes. Sometimes the chromosomes don't divide correctly during mitosis or meiosis. And if there are any mutations of the chromosomes, is one abnormally short? Is one abnormally long? Does one, only have a centromere in either of its other components? Karyotypes allow us to visually inspect the chromosomes for abnormalities, but it can't tell us anything about the genes on those chromosomes. So this might be a little small, but here we have a couple different karyotypes. You can look at these in the copy of the slides you have also. You can see that there are slight differences in each of these. So this second one up here on the right, we have the X and the Y. Perfect. So it's a male, but if you look above it, right above it to the right, you can see 3 little chromosomes for 18. We shouldn't have 3 chromosomes, we should have 2. So right there, you see that there's a small issue. If you go down, this person is XXY, so they have 2 copies of an X chromosome. You should only have 2 sex chromosomes, 2 X's, or an X and a Y, but this person has 2 X's and a Y. So as you can see here, they can tell us some things, but they can't tell us all of the things. So from this lesson, you should be able to tell me what a chromosome is and what is its role in cell division, what a chromosome is composed of, what is a karyotype and what it can tell you about an organism, and how are chromosomes different in a haploid and diploid cell. You can always use these slides for reference or this video. There is also an associated reading to go along with this. Thanks.